Welcome everyone to webinar. It's my pleasure to open this series of webinars, which is a cooperative effort between GlycoNet and the American Chemical Society Carbohydrate Division. So I would like to start with an introduction, but before I do that, just wanted to acknowledge that the University of Alberta, where the GlycoNet HQ is located on Treaty 6 land, which is the traditional territory of many First Nations, Métis, and Inuit. Our first speaker today is Professor Lara Mahal, who is currently the Canada Excellence Research Chair in Glycomics here at the University of Alberta. And she's an expert in glycomics, and she's very well known for her pioneering work on lectin microarrays and microRNA regulation of glycosylation. She got her PhD with Professor Carolyn Bertozzi when Carolyn was still at University of California at Berkeley. She got her independent career started at the University of Texas in Austin. And after getting tenure, she moved to New York University, where she was a faculty member until she was recruited here at the University of Alberta in 2019. And as I mentioned, she's, well, actually I didn't. She's currently the Canada Excellence Research Chair in Glycomics, and I'm looking forward to hearing her talk today.
Today, I'm going to talk to you about exploring the role of glycosylation in infection and immunity. And before I get started, I want to thank Warren for a lovely introduction. I also wanted to point out that we have a Meet the Speaker event next week. So if your friends couldn't make today and they want to see the webinar, it will be up online. And they'll have a chance to ask questions, and you'll have a chance to ask more questions as well at the Meet the Speaker event next week. Let's get started. So my lab really focuses on systems-based approaches to understanding the glycome. As Warren mentioned, we were the first group to ever create lectin microarrays, and I'll explain what those are in a minute for those of you who don't know. We use those with data integration to find a lot of information, and that's mostly what we'll focus on today. And we've also have a part of our lab that's working on a new hypothesis, which is the microRNA prophecy hypothesis, which I'm not going to talk about today. So today I'm going to focus on our work with lectin microarrays and data integration and how they tell us something about infection and immunity. But first, I'm just going to give you a brief primer on what lectin microarrays are. So this is from our very first paper in Chem Biochem. So lectin microarrays are quite simply a microarray of carbohydrate binding proteins, or lectins. So each of these would bind a different carbohydrate We fluorophore label the samples on the protein backbone. We bind those to the arrays, we then scan, we get a glyco pattern, and depending on what lectins light up, we can actually back out a molecular level analysis. And the reason for that is that really lectins bind very discrete molecular substructures. So, you know, I'm a chemist by training, and hopefully a fair number of you are also chemists, and some of you are biochemists and biologists as well. But from a chemical perspective, you know, these carbohydrate binding proteins are really quite specific in terms of molecular landscape. So for example, SNA, which is a lectin from Sambucus nigra, really binds to 2,6-sialic acid on a galactose. And so it has a very specific subspecificity. And when it binds, you know, when it is lighting up, it's binding this specific epitope. And I just want to remind you, you'll see a lot of the symbolic nomenclature for glycomics shown on the screen. But for those of you chemists, it actually does represent chemical structure. It's just a shorthand, because this is really hard to interpret when you get a lot of them in a row. Okay. So current lectin microarray technology. So in my laboratory, we currently do about 24 subarrays per slide. So on a single slide, we have a whole bunch of subarrays, each of which can do a separate sample. We actually use a two-color approach. So this is a combination of, actually, I'll move that over. I don't know if you guys can see that, hopefully not. Sorry, okay. So it's a combination of, so our two-color approach uses a sample and a labeled mixed reference. What's nice about this is it actually gives us an indexed binding so that we can mix the samples, bind to the array, and because of this, we can get really robust semi-quantitation. This makes it integratable with lots of other omics, and we have about 100 lectins and antibodies on the array, and we're constantly expanding this. So if you have more lectins you'd like us to include, we're always happy to take additional lectins to expand the capabilities of our array. So, okay, and for some reason, okay. So lectin microarrays, are really important in terms of being able to get really rapid glycomics. And so I'm going to tell you a couple brief vignettes of things that we've done in the lab. So we've used them, for example, to look at cancer. So in the area of melanoma, we've actually identified core glycosylation as being really critical in melanoma metastasis. And we've gone on to show that that actually impacts metastasis in a mouse model. So in this case, if you downregulate core glycosylation after cells have disseminated, so you're already in sort of a post-early metastasis state, you get a lot less cancer growing in the mouse. And so it really is not just an epitope that comes up because of being a marker, but rather a truly functional epitope, something that has a real impact driving human health, in this case, melanoma metastasis. So, and another example in women's health, looking at vaginal immunity, the vaginal cavity has lots and lots of mucosa, lots of immunoglobulins. These are all very highly glycosylated, microbial peptides, mucins. And when we look at that glycosylation and try to understand it in the realm of antimicrobial activity, we notice that cervical vaginal lavage from women that actually enables antiviral activity is high in levels of High-mannose. And this is very important because we also found in other studies that High-mannose is decreased when you have microbiome changes. For example, when you have bacterial vaginosis. And bacterial vaginosis is one of the epidemiological sort of negative signs for getting HIV. So if you have bacterial vaginosis, you have a lot higher probability of contracting HIV. And so we think that these glycoproteins that are given out by the vaginal cavity may have something to do with that, and that this High-mannose may be a marker, or indeed may play a role in the anti-HIV activity. So, but what I'm really going to focus on today is flu. So flu season is starting to come upon us. Given that all of us have been really dealing with COVID and coronavirus right now, I know that this is the next level of fear. It's like, what happens when we have the flu as well? And so the flu is a major issue, every year. So, originally people were comparing the coronavirus to the flu. And part of that is because the flu is actually quite deadly on its own. So I'm going to show you work that was done in collaboration with the FluDyNeMo group. This is a group of people who work together in systems biology and the flu, and their interest really is in trying to understand the host response. And it's headed by Elodie Gedden, who was at NYU, but is actually now at the NIAID. Bin Zhang is a bioinformaticist. Ted Ross has done all the ferret work. Alison Calvin did some of the virology work, and then Stephanie Bissell did a lot of the histology. And Elodie, of course, is a viral genomicist and really led this entire thing. So it turns out that in the original incarnation of the FluDyNeMo, they weren't planning on including glycans. And so why would you care about glycans in flu? Well, those of you who are carbohydrate and glycan specialists, which is probably most of you, may know that glycans are really critical in influenza pathogenesis. So sialic acids are the critical receptors of the influenza virus. And so they bind to sialicides. So if they're binding to 2,6 sialicides through the hemagglutinin, they are binding most likely to humans. If they're binding to the 2,3, they're binding to the avian and or swine. And so this actual change in sialic acid specificity is one of the things that allows avian flu to actually eventually passage to the point where it can infect humans. We also know that host protein glycosylation can act as a defense. The mucins are all highly sialylated. It's thought that that's one of the reasons why neuraminidase is required for cleaving through the sialic acid on mucins and releasing particles from the mucosal layers. And we also know that viral glycosylation interacts with the innate immune system. However, the majority of glycan knowledge to date was really focused on sialicides. So the question that we had was, well, okay, is that relevant to host response? We've only ever really looked at sialic acid for the most part in flu, but does that have any impact on severity, and what happens to the host? Because one of the things that we have to remember about flu, much like coronavirus, it has very different impacts in different populations, right? Flu can be very deadly to the elderly and to the very young. It has, whereas in the middle, we often have, you get very sick, but it's very rare for you to get pneumonia and die. We know that the virus is not different necessarily between populations, but the impact of it is. And of course, flu varies from season to season, and then the virus itself can actually change to be higher or lower severity in the population. But at the same time, even when you have a high severity virus, it doesn't impact all people evenly. So what is it about the flu that really, what is it about us that really changes how the flu is impacting us? Why do some people end up with severe flu and other people, it's a horrible week or two, but you're fine, right? So to look at this, we used a ferret model, and this is a model that Ted Ross uses at University of Georgia. And we infected the ferrets at day 0 with the 2009 H1N1 virus. This is the swine flu that came out in 2009. We then measured weight loss and collected lung samples at day 8. And we looked at uninfected control, mild infections, which were classified by a weight loss less than 10%, moderate infections, where the weight loss was between 10 and 16%, and then severe infections, where these animals had severe weight loss above 16%. So this is what the lectin microarray analysis looks like. So this is a 2-color microarray where we're doing each sample against all of the samples in the set. And if we take a look, you can see that it's a little bit hard to interpret from the heat map. But if we look at sialic acids, because that's what people often look at first, what we notice is there's no difference in the 2,6 sialic acids in the lung between uninfected, mild, moderate, and severe. There just isn't really any change at day 8. And day 8 is right at the point where you're at your severe point and starting to a little bit recover. Okay, so this is a place where you really are kind of at the low point right before the rise in getting better. If we look at 2,3 sialic acids, we can see that there is a decrease. This is actually pretty consistent with neuraminidase. Neuraminidase, even though the sialic acid that you bind to in the lung is in the upper respiratory tract by hemagglutinin is 2,6, most neuraminases are actually slightly more specific for 2,3. And so we actually do see a release or a loss generally of 2,3 sialic acid. But one of the things that you notice is that there's no correlation with the severity, right? There's a little bit of a loss, but it doesn't really have any significance in terms of what makes it severe. But then if you look, you can see that there's something growing in here. And if you look at this more carefully, it's actually the lectins binding High-mannose. So what we're seeing is a signature of High-mannose. So Griffithson binds, man 7, man 8, man 9. HHL binds man 7, man 6, and man 5. And what you see is they overlap in the High-mannose region, and you really see this high level of High-mannose coming up. And you notice that it's now dependent on severity. So people who have more severe, or actually in this case, I should say ferrets who have more severe flu are seeing more of a rise of High-mannose in their lung. Just to see whether that's actually true when you look at it histologically, we stained with HHL. So this is what an uninfected lung looks like. You can see that you have lots of air. You can see that you have lots of air spaces here. This is what you look like if you're infected at day 8. You're now compacted. You now have a lot of HHL, which is red staining, showing up. And so there is indeed a lot of this High-mannose response. If we look at this and compare it to the alveolar severity score, that's a histological score, we notice that the HHL binding score correlates with alveolar severity. So, okay, that's ferrets, but what about people? Is this at all relevant to people? So we looked at A549, which is a human lung adenocarcinoma, to see whether or not this response might be something that we could recapitulate in cell culture. So when we treat A549 with H1N1 and we look for the High-mannose, what we see is indeed HHL binding is found on the cells that actually have High-mannose. And so here you see So that actually have influenza. So here you see these are the influenza infected cells. This is the HHL binding, and you can see that these things come up together. If you treat this with mannose to inhibit the HHL, you can see that it is indeed inhibitable. The same is true if you treat with endo-H. So just to show that this is actually at the cell surface, we did a flow cytometry experiment with hemagglutinin. So these are A549 cells treated with PR8, which is an H1N1 strain. And these are control cells. You can see that there's no hemagglutinin. This is plus PR8. You can see that you have a cell population that has hemagglutinin coming up. And then this is, of course, PR8 with endo-H. Again, you have a population with hemagglutinin coming up. And what you see is that HHL binding is highest when you have infection. And if you then gate for only the cells that have PR8, that have HHL, that have hemagglutinin. So these are the cells that actually were infected, because not your entire population isn't necessarily. What you see is that the fluorescence in the HHL fluorescence, so the high-mannose fluorescence in those cells, is much higher and is indeed cleavable with endo-H. And this is 5 different replicate experiments. So if we then look at, so A549 is great, but it's a lung adenocarcinoma. So what about more sort of normal human cells? Will we see the same response there? So if we look at a primary bronchial epithelia, what we see is, again, we indeed get infection and we get high-mannose binding. And you can see here, again, the high-mannose is localized at the cell surface. You can see this beautiful co-localization of infected cells with high-mannose. And it's not strain dependent. So here's PR8, which is an H1N1. Here's H3N2, which is a different strain of influenza. And both of these, this is a flu A, here's a flu B. And in all cases, we see this high-mannose response. So what is this high-mannose response on? Well, we don't know the exact proteins that it's on, but what we do know is it's not just a result of the neuraminidase or the hemagglutinin, which are the 2 main glycoproteins on influenza. So if you look at lectin blotting, you can see that yes, the neuraminidase definitely has a lot of high-mannose, but there are a lot of other endogenous proteins that are coming up that have high-mannose increased on them as well, indicating that while this may be partly due to the neuraminidase expression at the cell surface, it's also due to a large amount of high-mannose being on other endogenous proteins on the cell. So why do we care about this? What is the role of high-mannose in immunity? High-mannose is normally thought of as an internal epitope, something that you don't normally see, although it is observed in stem cells in some cancers. And interestingly, if you look at the innate immune lectins in the body, most of them actually recognize high-mannose. And I want to call your attention to one in particular, which is the mannan binding lectin or mannose binding lectin, MBL. So we focused on MBL because MBL is actually known to bind influenza. So MBL, what is it? It's part of the complement-mediated pathway. It's the lectin complement-mediated pathway. When you get MBL binding to a cell or to an invading cell, for example, like yeast, you end up with an MBL MASP complex that then activates complement activity. Okay, it's the alternative complement pathway. We know that MBL binds flu. We also know that, interestingly, even though one would think that MBL would be protective, that the knockout mouse actually had an anomalous finding, which is that it lowered the severity of flu observed in the knockout. And no one really understood why that was. Interestingly, in human populations, increased MBL2 and MASP2 is seen in schizophrenics and multiple sclerosis patients. And both of those patient sets actually have much higher death rates from influenza and pneumonia. For schizophrenics, it's a 7-fold higher death rate. And for multiple sclerosis patients, it's 2.7-fold. Patients in critical condition with H1N1 were also found to have higher levels of MBL2 correlate strongly with death. So clearly, high levels of this mannose binding protein are correlating with severity of the disease. But again, that mechanistic link, like why that is not at all understood. So we wanted to see, does it recognize infected human cells? Is it possible that the mechanistic link is the upregulation of high-mannose on the cells of the lung when you have influenza infection? And so indeed what we see is that infected cells do indeed bind to MBL2. You can see that here. And if you treat with endo-H, you now do not see binding on the infected cells, indicating that the MBL is indeed recognizing high-mannose on infected human cells, which may help to explain the severity. So influenza severity is kind of a funny thing. It's actually, as I mentioned before, age-related. Infants, young children, and people greater than 65 years old tend to develop much more severe influenza. And about 75 to 85% of influenza deaths are in people over the age of 65. However, the 2009 H1N1 was actually anomalous in that older children and adults were more impacted. For older people, for those who are greater than 65 years old, this was thought to be due to pre-existing immunity. But the question remains, what underlies the age-dependent severity of influenza? So to look at this, we started to compare age differentiated ferrets. So obviously you can't do these experiments in humans. So what we looked at were newly weaned ferrets that mimic an infant population. So these are 6 to 7 weeks old. The lungs of the adult ferrets, which we had looked at before, and the lungs of aged ferrets who are 5.5 to 7 years old. And if you look at the ferret lungs across age, you do indeed see distinct differences. And so the first thing that I will note is that there's actually a difference in 2,6-sialic acid in the lungs of ferrets. So newly weaned and aged ferrets actually have slightly higher levels of 2,6-sialic acid than adult ferrets do. Whether that has an importance in terms of levels of infection, we don't really know, but it does actually but there is a difference in the sialicides in the ferret lung with age. We also see that, interestingly, 2,3-sialic acid actually goes down with age in the ferret lung. So we see a loss in the adult from newly weaned to adult, and even fewer of it in aged. And then the mannose is kind of interesting, because we actually start to see a separation of mannose epitopes. I mentioned that HHL kind of binds man5, man6, man7, and Griffithson binds man7, man8, man9. Well, that's because mannose is not mannose is not mannose. We, in our lab, define oligomannose as man5, 6, and 7, and High-mannose is 7 and above. And what we see is that the oligomannose portion, here indicated by GNA, you actually see a significant increase in adult and aged ferrets. But in the High-mannose portion, you actually go down a little bit in the aged, which is sort of interesting. Okay, but what happens with infection? So in ferrets, aged ferrets actually have extremely severe symptoms with the 2009 H1N1. They have high levels of weight loss and high levels of death. Now, the reason that they think that there is that aged people did not end up dying of the 2009 H1N1 in as high of rate is actually because they think that there was a similar virus many years ago that caused immunity to this new virus. So it's not a matter of them having different reactions. It's a matter of their immune system knocking it out very quickly. So if we look at an aged ferret population, aged ferrets really do show extremely severe reactions. And so now, because their weight loss is so severe, we actually cannot look at weight loss as a good metric for severity. And indeed, we went to a histological metric called a pneumonia composite score, which was set out by Stephanie Bissell and colleagues in this American Journal of Pathology paper that came out. So what we did is we quartiled them. So mild infections were considered a pneumonia composite score of less than 8.5. Moderate is in the middle, and severe is above 8.5. And we looked at their glycosylation patterns upon infection. And now for time, I'm not going to go through all of the other things, but what I am going to show you is that you can see here's mild, moderate, severe. You can see a real pneumonia composite score dependent, severity dependent increase in High-mannose, as indicated by Griffithson. And if you look, you'll also notice that there are a couple of other High-mannose lectins here, but also here's HHL and here's GNA. So we're actually also seeing it in this case in the oligomannose cohort. So mannose in general is really going up with severity in pneumonia, with severity in influenza, in the aged ferrets where we're seeing the severe impact. If we look at the young ferrets, so the newly weaned ferrets, these ferrets have very few symptoms. They're very mild. They have little death observed. Again, because we're dealing with growth rates of ferrets, weight is a little bit of a difficult metric to actually use for this ferret population. So again, we turn to the histological pneumonia composite score to separate them into mild, moderate, and severe. Now, interestingly, in these ferrets, and you can see it here, so this is actually a time course, what you see here, here's the data, there's almost no difference between mild, moderate, and severe, and if anything, it's slightly decreased from what you see in the uninfected. So there's no significant change upon infection in newly weaned ferrets, which very much goes with what we're observing in terms of the severity in this population. So with that, I'll leave you with what we think is happening. We think High-mannose is really a danger-associated molecular signal in influenza, and that the influenza virus is triggering this. I didn't tell you about the mechanism we think is existing here with IRE1 for the sake of time, but we do think that it's through this pathway, and we have some evidence for that. And what we think is happening is that this is then expressed at the cell surface and is recognized by the mannose binding lectin and probably other mannose binding lectins as well, but this is the main one we focused on, and that this then activates the complement cascade in inflammation, and that's what's leading to severity. When you have too much High-mannose or too much MBL2, we believe that you are then set up for having a very severe immune reaction to the flu that ends up causing additional damage and eventually death. So with that, I have to acknowledge the amazing lab who did all of the work, especially Shiwei Chen, who did all of the work that I showed you with the old versus young ferrets, Mohammed Eliash in the Hopman lab with Joaquin Lopez Orozco did the flow cytometry for us. Daniel Heindel did a lot of the work with the MBL. And this is, I put people's names on there, but these are the graduate students who were involved in the initial FluDyNeMo project. And of course, Elodie Gedden and Ted Ross, and all of my collaborators and all of you for your attention. And I'm going to open it to questions.